Hey guys, today we're going to talk about the nervous tissue as part of the series on IMAT biology preparation videos. Here we can see a picture of a neuron with its different parts. However, we're going to see them in more details. However, the main concept that I want you to get from here is that we have two main cells in the nervous system, the nervous cells and the glial cells. Now, the nervous system is divided in central nervous system and peripheral nervous system, and this is an anatomical distinction. The central nervous system is confined in the brain and the spinal cord. It's made up of, by the brain and the spinal cord, and it is confined in the skull and in the vertebral column. We can see that it is such an important and vital part of our body that it requires strong bony protection. The peripheral nervous system is the rest of the nerves in our body, as you can see, which is all over the body. All the nerves constitute the peripheral nervous system outside of the brain and the spinal cord. Now, the peripheral nervous system is functionally divided into the autonomic nervous system and the somatic nervous system. The somatic nervous system communicates with sense organs and voluntary muscles. So that means we have sensory input and motor output. So information that go into the central nervous system are called sensory, and information that exit from the central nervous system are motor, because they control sensory and motor. For example, if we see something, that information travels from our eye inside the brain. This is a sensory pathway, so it travels through afferent fibers. However, when we need to move a muscle, the information comes from the central nervous system, from our brain and the spinal cord, outside to the peripheral nervous system with efferent fibers. Now, the autonomic nervous system is important for the regulation of our internal organs, and when we mean regulation, and we call it autonomic because we do not have any control over it. Again, here we can see the main divisions. The central nervous system is made up of the brain and the spinal cord. Of course, we also have the brainstem at the base, at the top of the spinal cord. And the peripheral nervous system has somatic and autonomic fibers. And the autonomic is divided into sympathetic and parasympathetic, as we will see. Now, the central nervous system, again, is made up of the brain, the brainstem, and the spinal cord. And then the peripheral nervous system has the somatic, which is for voluntary muscle movement, but also for sensory input, and the autonomic, which controls functions that are autonomously controlled in our body and are beyond our control. For example, our heart rate, our respiratory rate. Now, in the nervous system, we have two types of cells, the neurons, which are the main cells who serve the purpose of the nervous system, which is transmission of information and integration of that information and decision making, and the glial cells, which are supporting cells. They protect the neurons and they help them function. We have many types of glial cells. The most important to know is the Schwann cells and the oligodendrocytes, which are responsible for the myelination of our neurons. What does that mean? The same way a cable is protected by some material on the outside for insulation, so electricity is not lost, and the transmission speed is faster. The same way our neurons are covered by myelin, which is fat. A type of fat, and this serves the purpose of insulation and gives a faster transmission speed in the information. Then we have astrocytes and microglia. Microglia are macrophages, which are found in the central nervous system. and then we can proceed to see a little bit more about myelination. But it's important to know that Schwann cells are responsible for the myelination of the peripheral nervous system, and of course the neuron axons, while the oligodendrocytes are in the central nervous system. Now, here you can see a nervous cell, and we should start and describe the main areas. First, we have the soma, where the nucleus is located. Then, before the axon starts, is this area, the axon hillock, which is a very, very important segment. And the axon is responsible for the transmission of the information from through distances. And then we have at the end the axon terminals. So the way this information would be transmitted is like that. So we have a unidirectional transmission of the information. The dendrites would receive information, would then pass it on into the soma. Information from the different dendrites, either positive or negative, will be integrated at the area of the axon hillock. Then it will travel down the axon. The information travels in one direction, where it will reach the axon terminals. Then the axon terminals are going to communicate with dendrites of the next neuron, and so forth. This is how the transmission happens. And here we can see that the membrane of the nervous cell is called axolemma, and the cytoplasm of a nervous cell is called axoplasm. And here we can see the Schwann cell surrounding the axon, providing insulation with this myelin sheath. Here we can see how that happens. The Schwann cell, it is a normal cell that starts to wrap around the axon. And the way it does that, it leaves very, very small segments of only membrane which surround the axon. So the myelin sheath is basically a lot of plasma membrane that has turned many, many times around the axon of a neuron. Now this is each Schwann cell surrounds only a small part of the neuron. So one axon of one neuron would have many Schwann cells, while in the central nervous system, the oligodendrocytes have many processes that come and do the same process as the Schwann cell. However, one oligodendrocyte can myelinate many neurons, while here, in the peripheral nervous system, one Schwann cell will myelinate one part of one axon. Now, the neurons, we talked about the soma and the axon and the dendrites. However, we need to understand that we have different types of neurons with different morphologies. We can see this neuron that has a very complex tree of dendrites. That means it is receiving a lot of information. We can also have unipolar neurons, which have a peripheral process and a central process. And what does that mean? That means that the signal will come from the central process, it will go up in the soma, it will be integrated, and then it will be sent into the peripheral process. This is the transmission in a unipolar neuron, and that's why we call it a central process and peripheral process, because the central process comes from the central nervous system, and the peripheral process usually exits in the peripheral nervous system. We'll see an example of a unipolar neuron when we talk about the spinal cord. And then we have bipolar neurons, which have dendrites and then one axon. And then we can see the multipolar neuron when we have many dendrites and one axon. And again, there are many type of neurons, as you can see. However, I want you to notice that each area of a neuron has a specific function, and this is what is responsible for the unidirectional transmission we talked about. So in the beginning, there is a region that is responsible for the input of the information. Then there is an area which is going to integrate the information, the so-called axon hillock. Then the conduction, which is basically just transferring the information in order to give off output to another neuron. However, this output cannot only be to a neuron, it can also be to a muscle. It can also be to a blood vessel. So this is important to understand. Now, we can see that this is a cross-section, a diagram of our brain, our brainstem, and our spinal cord. And you can see that if we have a neuron which has its dendrites at this level, and it has its peripheral process or its terminal output centers here, then this is going to be a descending fiber. Why? Because the information descends from the higher levels of the brain down to the spinal cord and then to the periphery. While if we have the opposite, that means that it is gonna have the dendrites here and the terminal output area here, then this is gonna be an ascending fiber because the information ascends from the periphery to higher areas of our nervous system. And information that enters the brain system is sensory information or afferent, while information that exits our brain and our central nervous system is motor or efferent. And this is a simple reflex. No, not only reflex, but a simple pathway, let's say, a neuronal pathway, where we feel something, the information goes with a sensory fiber. You can see dendrites here. So it senses through this area. Unidirectional transmission to the terminal buttons of the neuron. Then interneurons, there are different neurons that are going to integrate the signal in different ways. And then we have the output, a descending fiber, or a motor, or an efferent fiber, which is going to be responsible for passing the signal to the muscle. And there are thousands of pathways, or even million pathways like that, which allows us to act and behave and move by sensing our environment and then responding accordingly. So again, this is a reflex. What does a reflex mean? A reflex means that we do not have any control over it. It just happens. So there are two major steps in a reflex. One is the stimulus and the perception of the stimulus. So the stimulus is here. However, a neuron, some dendrites, or a sensory organ needs to be activated, and then this is going to travel inside our spinal cord, and then it's going to exit immediately to activate a motor neuron. For example, we pull our finger when it touches, or a hand when it touches a sharp object. This is a reflex. However, we know about this because it sends collaterals up to the brain. These are ascending fibers, therefore, because from the spinal cord they go to higher centers, because our conscious areas are in the brain, in the higher levels of the brain, so in the cortex more specifically. So the information needs to go there, so we are aware that we did the reflex. It's not just like we saw our hand move. If we were also blind, we would know that our hand moved like that, because there are also fibers that go upstairs, let's say, that go to the ascending centers in the brain. Now, you can see here that there is an interneuron, so that means a neuron that is between two main neurons, and this interneuron is not mandatory. Sometimes we have reflexes that are only between two neurons, and we're going to see some of that a little bit later. Now, the nervous system behavioral control. That is the autonomic nervous system, or the visceral nervous system. We have no voluntary control over this system, and it has two main functions, two main divisions, the sympathetic division and the parasympathetic division. A very easy mnemonic to remember the different functions of the two divisions is that the sympathetic nervous system is the one nervous system that is activated for a fight or flight response. That means we either stay and we fight the enemy, or we run from him. That all the actions that we must do in order for that to take place is the sympathetic nervous system. While the parasympathetic nervous system is the so-called rest and digest. So when we're resting and we're digesting, it is the parasympathetic nervous system that is performing these functions. I want you to understand that there is a balance between those two systems, these two divisions, because many times they are competitively regulating the same organ, uh, in a different way. Let's see an example. For example, if we can see our heart rate, the parasympathetic nervous system slows the heart rate, while the sympathetic nervous system accelerates the heart rate. So we can see that there is a balance between the two nervous systems. Sometimes the sympathetic wins, sometimes the parasympathetic wins. When the parasympathetic wins will be when we are at the rest and digest. When we are chilling, watching Netflix, then we're resting and digesting. So we have a slower heartbeat. While if I go running, I will have a higher running because my sympathetic gets activated. What does that mean at the same time? At the same time, it means I need oxygen. If I need oxygen, I need to dilate the bronchi so I get more air inside. The bigger they are, the more air they are allowed to pass through. And also the lower the resistance is. So I need to breathe more. I am hyperventilating, let's say, when I am running. So this is the same function. And at the same time, I need to produce glucose. Why? Because my muscles need some fuel in order to run. So my liver needs to convert glycogen to glucose in order to provide fuel for the muscles. And at the same time, the parasympathetic nervous system, and I'm sorry, one more thing. About the digestive system, we can see that it inhibits the peristalsis and the secretion. So basically, it's saying to the digestive system, you know what, it is not your time to shine right now. Now, we don't really need to digest, we need to make sure we survive. While in the rest and digest phase, we stimulate peristalsis and secretion over here because it is the time to do those things. We're chilling, we're watching Netflix, so we are resting and digesting. And also, you would hate to need to urinate while you are running away from the enemy. So at the sympathetic nervous system, we inhibit the bladder contraction, while in the parasympathetic nervous system, we contract the bladder. The main neurotransmitters responsible at the level of the parasympathetic nervous system is acetylcholine, while in the sympathetic nervous system, we have adrenaline and noradrenaline. So when you say, oh, I had such an adrenaline rush, it is actually your sympathetic nervous system being activated. Also, when you're anxious, when you're nervous, you have a sympathetic system hyperactivity, let's say. It wins over the balance that we were talking about earlier. Now, again, we can see a more simplified explanation of all that we said. The sympathetic nervous system, fight or flight, we dilate the pupils, we need to be able to see better, inhibit salivation, increase the heartbeat. You're saying when you're nervous, you say, I have such a fast heart rate, I'm breathing so fast, we increase our respiratory rate. And you also say, my mouth is so dry because the sympathetic nervous system inhibits your salivation. While in the parasympathetic nervous system, everything is fine. You breathe in a normal heart rate, your pupils are constricted, you have enough saliva, and you stimulate the activity of the intestines because it is rest and digest. While in the fight or flight response, we inhibit the activity of the intestines and also of the blood vessels. So the blood vessels of the gastrointestinal nervous system, they have specific receptors that when the sympathetic neurotransmitters are released in the blood from the adrenal medulla here. Here is the adrenal, an organ that is on top of the kidneys. It is a modified sympathetic ganglion, as it's called, and that just means that instead of the neuron, okay, let me explain a bit better. We have seen so far this model, that there is a neuron that secretes neurotransmitter to a dendrite of the next neuron, right? And that here would be a synapse. However, sometimes this is, and where we have bodies like that, these, okay, the axons, the collection of axons are called nerves. So what are nerves? Collection of axons, many axons together. When we have many bodies together, we call that a ganglion. So usually we have synapses where we have bodies, so inside a ganglion. So a ganglion is just where there are an area that there are many bodies or soma of the neuron. Now, it is called a modified sympathetic ganglion because instead of having this over here, what they do over there in the adrenal medulla is they secrete their substances directly into the blood. This is the blood. So they secrete the substances directly here. And the neurotransmitters that they secrete, we talked about adrenaline and noradrenaline. So not only will I secrete these, for example, immediately in the heart or inside the lungs through these nerves. You can see right here, there are these nerves that come off from here and they innervate the heart, they innervate the lungs, they innervate all of that. However, they also go to the adrenal medulla where they secrete those substances in the blood. So they can act on different blood vessels. Blood vessels in different areas have different receptors. So the same molecule that they secrete, adrenaline, for example, or noradrenaline, which are also called epinephrine or norepinephrine, same substance, different names, are responsible. So same substance, different receptor, that means different action. So in a blood vessel that is in the airways, for example, the adrenaline is going to cause it to dilate. We need more blood going there, right? More blood getting oxygenated. Why? Because it has a specific receptor that will respond to the adrenaline in a specific way. While in the intestines, one blood vessel is going to have a different receptor that is going to respond to the same substance, adrenaline, in a different way. It's going to cause vasoconstriction. Well, this is constricted now. So we have the vasoconstriction here because we don't need our blood going to the intestines when we're fighting or flighting. Okay, we need the blood to go to the muscles. We need the blood to go to the heart. We need the blood to go to the lungs. Okay, so this is maybe a little bit too detailed, but I just want you to get some main concepts that we have nerves. We release a neurotransmitter to the nerves at different areas that are responsible for functions. So for instance, we have nerves here. We're going to release a neurotransmitter to those nerves, and those nerves are going to inhibit the activity on the intestines because they innervate a muscle, for example. Sometimes, though, we have nerves that secrete nerve cells that secrete their substances directly into the blood. And that's how we get adrenaline and noradrenaline in the blood. Now let's move on. How does the autonomic nervous system work? With reflexes. What does that mean? That means that if we have a high blood pressure, there's going to be a sensor, a sensory organ over here that's going to sense the increase in your blood pressure. It's going to travel through a nerve into the brainstem, and then through a small reflex, you are going to get an effector nerve fiber that is going to immediately innervate the cells in the heart that are responsible for the heart rate, and they're going to decrease it in turn. So all of this is beyond our control, and hence the name autonomic. It is called autonomic for a reason, and that reason is because we don't have any control. They are reflexes. You see, we don't even get a collateral. We don't even get a collateral to go up here in the cortex where I would sense that. I don't know that my blood pressure is up. I don't know that my heart rate went down. I can feel it. But this is a different. This is the somatic nervous system that we were talking about earlier. Okay. You don't have information about your blood pressure, about your heart rate, about your respiratory rate. You need to consciously investigate in order to get information about those functions. And that is the whole essence of the autonomic nervous system, that it is beyond our control and we do not know about it. The brain does what the brain wants, and it's not exactly the brain, it's the nervous system. How do we control the autonomic nervous system? Well, we're going to see this a little bit later. Now, we're going to switch to an entire different topic of conversation, and this is, we are talking about transmission of information and electricity, but we're going to go into more detail to understand what we mean by information, how is information coded, and why we're talking about electricity. But first, let's go to some very, very fundamentals to know that in the cells, there is a low sodium concentration inside the cell. I here means intracellular fluid, and here it's extracellular fluid. So the sodium is very low, while the potassium is very high inside the cell. While outside of the cell, in the extracellular fluid, the sodium is very high, and the potassium is very low. So we see this difference in the concentration. So we see that there is a concentration gradient for sodium to enter, right? That's what the sodium wants to do. If I leave the sodium alone through channels, the sodium will want to enter. And what does the potassium want to do? The potassium wants to enter. So at some point, the concentration will be equal. Now you're wondering, why aren't the concentrations equal? Well, because we have pumps, and we have sodium, potassium, ATPase. What are these pumps? These are pumps that are responsible for pumping sodium outside and potassium inside. This is potassium. This is sodium. Remember, the sodium was higher on the outside, the concentration, and lower on the inside, and vice versa for the potassium. Why? Because of these pumps. These pumps are in the membrane. And also this pump uses ATP, energy, because it is actively transporting. We have active transport of sodium and potassium against the concentration gradient. Now, let's talk about the electrochemical gradient. Okay, if we talk about the electrochemical gradient, it has two main parts, electro and chemical. First, the electro. Electro means electrical potential energy. And we're talking about a membrane potential, which is VM, is how we usually write it down and call it, which is the membrane potential, which literally means difference of charge across the membrane. So that means that if it's not zero, that means one side of the membrane is more negative than the other. So we have some more positive charges on one side and more negative charges on the other side. And if we have, let me show you something. So this is my membrane, right? And I'm telling you the VM, the electrochemical potential, the membrane potential is not zero. So that means that there are more negative charges here. So this side is more negative because the VM shows us the difference in charge. And on that side, there are more positive charges, right? Now, if I place here a sodium, that was a bad example. Actually, let me delete that. I don't want to place the sodium here. If I place here the chloride, which has a negative charge, it will be attracted to the positive, so it will want to move towards that side of the membrane, right? So now we're not talking about concentration gradient like we have seen, and we're very familiar with diffusion and everything, but we're talking about electrical gradient. So this electrical gradient, because of the charge, the chloride, which is negatively charged, it gets repelled from the other negative charge and also attracted to the positive charge. So it will want to go over to that side of the membrane. Okay, and that is what electro means. Electro shows us how the charge influences the movement of different ions mainly. Chemical, on the other side, is what we know and understand, which is the diffusion. It's the concentration gradient, right? This is very simple. It's called chemical potential energy. So that means that if the concentration is greater on one side than the other, so let's say we have a high concentration, again, just a quick revision. We have a sodium concentration here that is 145 milliequivalents, while the sodium concentration here is 12. We see that the sodium will want to move down its concentration gradient through that. And that little arrow, that force that drives sodium to that side of the membrane, is the chemical gradient. So, for each substance, we can define an electrical gradient, so an electrical force, and a chemical force. Now, if I tell you that this is the inside, and this is the outside of the membrane, and I tell you that the membrane potential is given by the potential on the inside minus the potential on the outside. So, if the membrane potential is less than 0, that means that the environment inside is going to be more negative than the outside, or the outside is going to be more positive than the inside. Okay, and I also tell you that the concentration of chloride inside is lower than the concentration of the chloride outside. So if we give the force by electrical potential energy, then what happens? Then we can see that the environment inside the membrane, right? The environment inside the membrane is more negative, and the chloride is negative, so the chloride will want to go over to the positive side, to the more positive, not absolutely positive, but it is more positive than this side. We're talking about relevant concepts right now. While we said that the concentration of chloride is greater on the outside than the inside, that is wrong. I'm sorry. The concentration of the chloride is greater on the inside than the outside. So the chemical force will draw chloride again outside. Okay, if we add these two forces by vectorial addition, then we get one unified force, which is the electrochemical gradient. Now, how do we get the resting membrane potential? I told you that it tends the inside of the cell tends to be more negative than the inside. How? Well, first of all, I have chlorine, potassium, and sodium, and let's say we have equal concentrations for both on both sides. Well, we have some proteins which have a negative charge and some phosphates which also have negative charge that are inside the cell, and they cannot pass through the membrane, and they're only concentrated on the inside. So we see that we already have a more negative concentration on the inside of the cell. So it has gone a little bit towards the negative. So we have a little bit of resting membrane potential, which, let's say the VM is minus 10 millivolts right now, just because of that small difference. Then we need to consider the activity of the sodium-potassium pump. The sodium-potassium pump creates a sort of concentration difference between sodium and potassium. The sodium is going to be more concentrated on the outside and the potassium more concentrated on the inside. However, the sodium-potassium pump is also electrogenic. That means it creates membrane potential. Why? Because, I apologize for the noise, because it pumps 2 potassiums on the inside, while it pumps 3 sodiums on the outside. So if I remove 3 positive charges and I enter 2 positive charges, that means we're going to have a deficit here, 1 negative charge. And why? Because before we said everything was neutral, right? except the protonix and the phosphate. So that means all those sodium charges. So I had 1 sodium, 2 sodium, and 3 sodium. Let's put them like that. And they're all positive, right? I'm sorry, I'm going to put it on the outside. They were balanced by a negative charge. Now those 3 left. So for just a slight second, we have 3 new negative charges. However, then 2 potassiums enter. 1 potassium entered, so it balanced 1 negative charge, and then another potassium entered, so it balanced another negative charge. However, 1 negative charge remained unbalanced, so it is electrogenic. It makes it even a little bit more negative. And then we have a potential, a chemical gradient now that has developed for potassium and sodium that drives further this force. So let's check this out. Now, this potential, it also drives chloride from the intracellular fluid to the extracellular fluid. So as chloride leaves out, what happens? We get a little bit more negative, more positive. However, also a lot of potassium wants to drive out, even though some enters. So the potassium is balancing the leave of the chloride, but the sodium is going to enter as well. It is more complex. It is a much more complex process. However, you're going to learn more about it in the physiology when you go into medical school. However, I want you to have a general understanding how the ions, and there are specific 3 concepts that you need to know. First of all, the material that cannot pass, like proteins and phosphates that are on the inside. Second of all, the sodium-potassium pump, which contributes to the resting membrane potential, and then the movement of specific ions in specific ways according to the electrochemical potential that help develop that very negative resting membrane potential Vm. And when I'm saying very negative, for most neurons, it's at minus 70, minus 75, let's say, millivolts. Then, so this is the resting membrane potential at minus 70. Then we have a threshold potential, which is a value defined by the millivolts, by a millivolt number, minus 55, after which the neuron is going to shoot an action potential. An action potential has a rapid depolarization and repolarization. Why is it called depolarization and repolarization? These are concepts that the depolarization we have when we go up towards the millivolt scale, and repolarization when we go down. And why is that? Because when the cell has a millivolt that is at negative 70, right, that means the inside of the cell is at minus 70 and the outside is at 0 millivolts. Because the outside, we consider it infinite. It's the blood, everything. We consider that everything is balanced. So the outside is 0 millivolts and the inside is minus 70. And this is the outside. So in the outside and the inside, they're very different. So we say that the cell is polarized. The membrane of the cell is polarized because it has a positive and a negative charge on 2 different sides. However, if I cancel the depolarization, so I go towards 0 from the minus 70, so when I go towards 0, then I'm canceling that polarization. So that means I'm depolarizing my cell. While if I go down towards my minus 70 or more negative values, then I am polarizing the cell again. So I am repolarizing the cell. And that is why we call it depolarization and repolarization. Now, let's see it a little bit more in detail and understand what is action potential. So first, we have some local potentials that we're going to see again in a little bit that drive up my potential to the threshold value. That's all I want you to remember for now to understand. So I drive it to the threshold value. So my membrane potential is at minus 55. Then we have a depolarization, where what happens is we have an increase in a rapid increase in potential. It reaches about 120. Why is that? Because at this level 3, many sodium channels open, voltage gated sodium channels. Why voltage gated? Because they sense the threshold. When they arrive at -55, they say, okay, I'm opening now. So these voltage gated sodium channels open. And what happens? A lot of sodium enters the cell, right? Isn't that what sodium wants to do? Because both its concentration gradient and its electrical gradient. So the electrochemical gradient drives sodium inside, because inside is more negative, and also because we saw that the sodium-potassium pump creates that concentration gradient of higher sodium concentration on the outside and lower sodium concentration on the inside. Then, once the potential reaches values above 0, so after this level, potassium channels open. Potassium channels drive the potential down because the potassium wants to exit because of its concentration gradient, if you remember. The potassium wants to exit, and when positive charges exit the cell, what happens? Negative charges are left inside unbalanced, and as a result, the potential is repolarized. So much so that it becomes even more polarized than it was before, which is a state of hyperpolarization. And then we go back to our resting membrane potential. Now, in order to the neuron gets constantly many potentials, many local potentials. However, it needs to get enough of them to reach the threshold potential here. And this area that needs to reach the threshold potential is the axon hillock. That's why before we were saying that at the level of the axon hillock, we have the integration of the membrane potential at the level of the axon hillock, because it's where we have this threshold, these first voltage-gated sodium channels. And now in this slide, you can pause the video and take a look and read what was happening in the previous slide. And now we can move on. Here you can see a picture. So we have the resting membrane potential, then the sodium channels open, so the depolarization begins, and the potassium channels begin to open, but they're much slower. The potassium channels start to close and more potassium channels are fully opened. So the potassium leaves the cell and leaves positive charges unbalanced on the outside. So we have reached the level of 20-35 that I said before. And then we reach the lower level when we have the hyperpolarization. Now, I want you to focus on one main concept, the refractory period. The refractory period is the period in which the cell, the neuron cannot fire another action potential because one action potential is underway. There is an absolute and a relative refractory period. The relative refractory period means that with enough force, with enough depolarization, so the threshold is higher in the relative refractory period. So I can fire an action potential, but I have a very high threshold. However, in the absolute refractory period, it doesn't matter how much depolarization I do, because this is a small depolarization as well. It doesn't matter, I will not fire another one. But here, if I reach a high enough threshold, I can fire another action potential. And that is an important concept, because in the nervous cell, the action potential and the transmission of the signal, what is the signal? The signal is the action potential, and the action potential travels down the axon. That is very important to understand. Why? Because this little thingy that we saw, this one, This one happens here. It's going to happen again here, and it's going to happen again here. It's happening here right now. This is still in the refractory period, so that means if I want to be more accurate, now it's at this phase here. This one is normal. It has already passed. However, it happens in segments, so it goes here, the action potential, then here, and this is where we have the channels. And we're talking about electricity because we're talking about movement of ions, movement of sodium and movement of potassium. So movement of charged particles is electricity. So we have little action potential and changes in the membrane, reversal basically in the potential of the membrane, because you saw that we reached 35. 35 is positive from the minus 70 that I was at. So we have a reversal, then the positive becomes negative and vice versa. And this is a refractory membrane because it is already in that hyperpolarization phase. So we are at the refractory period, and that is important because here we can see the Schwann cells. At the level of the Schwann cells, we do not have action potential happenings. So that means that action potential can happen at this little gap that the myelination leaves, which is called nodes of Ranvier. This little gap is a node of Ranvier. So that means we have saltatory conduction. Saltatory conduction is when the action potential happens here, then it's going to happen here, then it's going to happen here, here, and here. And I want you to notice something. Here is the sodium entering. It's depolarizing this area, but it is also traveling down this direction, where it is going to reach that membrane. It is going to help it reach the threshold. So another action potential is going to be fired here, because this membrane reached the threshold. However, it can also go back. So another action potential could be fired here, right? Wrong. Why? Because this is at the refractory. This is a refractory. So the refractory period is important for the unidirectional transmission of the information. And you can see how it progresses. I know these are wild concepts and I'm going through them really fast right now. However, you do not need to know them in great detail. I just want you to understand the main concepts. Now, the same way in the axon, the information was going like that. In a synapse, the information is also going to go like that. We have unidirectional transmission of information at the level of a synapse. And what is the synapse? The axon terminal with another dendrite of another axon. So here we have the axon terminal, and here we have the postsynaptic neuron. And we also have a synaptic cleft between the two, which is a small gap. So we see the area that we need to travel is quite small. And let's start with that slide. Again, here you can see that we only see this small area, the communication between two nerve cells. Now, what happens here? That is very interesting to see. We said that we release a neurotransmitter. In this case, we're going to release acetylcholine. It is one of the many neurotransmitters. Acetylcholine is going to bound to a channel, which is called a ligand-gated channel, because once a ligand binds to it, the channel opens. Now, before we saw voltage-gated sodium channels, where they opened at specific membrane potentials. Now we're seeing ligand-gated ion channels that they open when a ligand, a molecule, binds at them. If acetylcholine is a neurotransmitter and the channel it binds is a ligand-gated sodium channel, sodium is positive, right? And it enters the cell. We saw the electrochemical potential. It wants to enter the cell. So acetylcholine is gonna induce the increase of intracellular sodium, and as a result, more positive charges. So it's going to cause a small increase in what? In the in the membrane potential, right? We were at minus 70, now we might have gone to minus 60. However, acetylcholine can bind to a different channel, which is a potassium channel. In that case, it is going to cause hyperpolarization because positive charges are going to leave out of the cell. So negative charges are going to be left unmatched, uncoupled. So they're going to now have a say in the memory potential. What does that depend? That depends on the postsynaptic neuron. You see, the presynaptic neuron is releasing the same molecule. What? Acetylcholine. Now, the postsynaptic neuron can have in its memory ligand-gated sodium or ligand-gated potassium channels. Okay, and acetylcholine is just an example. It's much more complex. However, I just want you to understand this concept that one presynaptic neuron can be releasing, can make many synapses because you can have another postsynaptic, presynaptic photon and another. But this can go to a neuron that has ligand-gated sodium channels, and this can go to a neuron that has ligand-gated potassium channels. This acetylcholine is going to activate that cell. Why? Because it is going to help it reach its threshold, because the sodium is going to depolarize the cell a little bit, and it's going to help it reach its threshold in order to shoot an action potential. While the other neuron that has a negative, the other neuron that has a potassium, a ligand-gated potassium channel, when acetylcholine binds to it and allows the free movement of potassium through it, it's going to inhibit the cell because it is going to hyperpolarize it. So it's going to get it further away from its threshold. And you can see here that we can have excitatory postsynaptic potential, which is what happens in the postsynaptic cell because it drives it closer to the threshold, or inhibitory postsynaptic potential that drives it further away from the threshold. And that's why we say that a neuron can either be excited or stimulated by another neuron, or inhibited, okay? Because of this concept. The concept that we have excitatory and inhibitory postsynaptic potential. And you can see that in order for a neuron to fire an action potential and to start firing an action potential, we need to have many excitatory postsynaptic potentials. And this is not even the true situation. The true situation would go something like that. So let's say this is my resting and this is my threshold, right? It would go a little bit up. You got an excitatory. Up, then you got an inhibitory. Then you got another excitatory and another excitatory, but then another inhibitory, and then another excitatory, and then another inhibitory, and then another excitatory. Oops, I passed the threshold. What happens now? The cell, the voltage gate sodium channels are going to open, and boom, action potential. Okay? Now, let's see what happens at the level of the neuromuscular junction. I'm talking about neuromuscular junction. It means that a nerve is innervating a muscle. So the neurotransmitter at the neuromuscular junction, which is the synapse, let's say, between a nerve and a muscle, is always acetylcholine. And this innervation is going to cause the contraction of the muscle. How? Well, acetylcholine is going to be released, and it is going to bind to an acetylcholine ligand-gated channel. And this is going to cause what's called the end plate potential, a potential inside the muscle cell, because the muscle is excitable tissue as well. Okay? And these channels actually allow both potassium and sodium, but it is more complicated than that. So let's say it drives an action potential inside the end plate potential, which is going to start traveling because it starts shooting action potentials. So the end plate potential is something like a postsynaptic potential that we saw before, and then we're going to have an action potential. Now, this action potential is going to travel all throughout the membrane of the muscle and also down the T tubules, which are invaginations of the membrane inside the cell. And once these action potentials go, they are going to induce the release of calcium from the terminal system of the smooth endoplasmic reticulum, or the sarcoplasmic reticulum, as it's called in the skeletal muscle. Why? Because we have some voltage-gated calcium channels there. So they sense this difference in the potential, and in turn they release calcium. So you can see that everything is electricity. And once calcium is released, we're going to have the contraction cycle, as you can see here, and with another summary here. And of course, we didn't say why we have the release of the presynaptic vesicle, which is a vesicle inside here that contains acetylcholine. Well, it's quite simple. The action potential arrives at the end of this axon. Once it arrives here, we have a voltage-gated calcium channel. Calcium enters the cell, and once calcium enters the cell, it induces the fusion of this vesicle with the membrane, and as a result, the release of the acetylcholine. Okay, this is in the presynaptic. Then we have here at the muscle fiber, where we have an action potential because of the opening, because of the end plate potential, which end plate potential is caused by the binding of acetylcholine to a ligand-gated ion channel. That end plate potential induces the drive of an action potential. The action potential travels down the T tubules, and as a result, calcium ions open, channel for calcium open. And we have the release of calcium intracellularly, which drives the contraction cycle. Now, what I want you to see is that we have many synapses inside the spinal cord. And in the central nervous system, we have areas called dark matter, like this one. All of that one here is the dark matter. I'm sorry. And all the outside is the white matter. And this one here. What is the difference? In here, we have synapses. We have bodies. Remember how in the periphery I told you that all the collections of bodies were called ganglion? Well, in the central nervous system, all the bodies are collected in the dark matter, while all the axons are collected in the white matter. And they form specific tracts, which is many axons traveling together to a common destination. While in the periphery, and the organization in the spinal cord is the dark matter is on the middle, the white matter surrounds, while in the brain it's opposite. The dark matter is on the outside and the white matter is on the inside. I might have a picture later on. While again, in the periphery, which is right outside here, we can see this very, very beautiful ganglion. In the periphery, we have ganglia, which are collections of cell bodies. This soma of the neurons. Now, how are these neurons? These are, as we saw before, the neurons that are with this polarity. They have a peripheral process, which receives information from the periphery, and a central process, which brings information into the central nervous system. And these are the sensory fibers. The sensory fibers have this organization. They bring the information into the spinal cord, and from the periphery, with their bodies being all of them together. These are unipolar neurons, by the way, with their bodies of all those unipolar neurons being in the posterior root ganglion, because there is a posterior root, which are the fibers that enter the spinal cord. But the roots that are going to leave from the anterior root, the fibers that are going to leave from the anterior root, these are motor fibers. They're going to go to innervate muscle. So that means they have this direction, right? They're going to go to innervate something. But their bodies, they're in the normal axis we have seen, and their bodies is over here. So this is the general organization at the level of the spinal cord. So you can see the motor pathway, and in this picture, the spinal cord is over here. So the fibers exit like that to go to the muscle, and here the information is taken from the periphery, and they have their cell body here, and then they enter the spinal cord through the posterior root. So the spinal cord has two roots, the posterior and the anterior. Again, here in the cross section, you can see that. You can see the posterior root ganglion, you can see the anterior root. So if I wanted to draw this better, let me take a different color for the motor fiber. That is an awful color. So it's going to travel with this direction. I'm sorry, it shouldn't go to the anterior root. Yes, I did a mistake here. Okay. They're going to travel like that. This is a sensory nerve, a motor nerve, I'm sorry. And this is a peripheral process. And they're going to travel like that inside with the spinal cord, and the cell bodies being here at the level of the posterior root. However, after that, we have another division. And here we have mixed fibers. It doesn't matter. So some of the motor might go there, and some of the sensory might come from there, and some of the motor might go here, and some of the sensory might come from here. But at this level, we're going to have the division of them into a posterior root, a sensory, and the motor into an anterior root. And of course, we can see that we have these meninges, which are also an important thing to mention, which are membranous covers of the central nervous system, and they contain fluid beneath them. The meningeal, well, depends where we're talking, but the so-called CSF, cerebrospinal fluid. Why? Because if you fall down and there was no covering, your spinal cord would hit bone, and that is dangerous. It could get severely traumatized. So it has this fluid that it floats in, and it protects it, and these membranous coverings, the meninges, which protects it. And also, it makes it much lighter. It makes your brain much lighter. Your brain is much more heavy than your whole head feels, and that's because your brain literally floats in the fluid. And the three meninges are the dura mater, the arachnoid mater, and the pia mater, and you should know their names and their order. Dura is more outside, arachnoid is in the middle, and pia is a very intimate lining of the central nervous system. And then you also have some fat in the epidural space. Why epidural? Because it's outside of the dura, which is the most outside lining. Outside, you have this epidural fat, which offers a little bit more of a cushion in case, I guess, you fall and or you just move around. Imagine your spinal cord constantly hitting your bone. That would be disastrous. Or your brain. That would be, again, disastrous. And again, this is the organization I was talking to you about. You can see that you have afferent information entering from with these unipolar neurons. So you have the peripheral process here, the central process over here. And as a result, this is the direction of information. The information comes inside. How? We saw how action potentials traveling, they get integrated. Here we have presynaptic postsynaptic potentials. They get integrated at the axon hillock. Then they travel all the way down the central process. Here they release I'm sorry, I had actually paused the recording and I forgot what I was talking about. So yes, we have the unipolar neuron. The information enters in this direction, enters like that, and we have an action potential traveling. It is integrated in the axon hillock, the peripheral process, then the central process. Then the action potential reaches here. It meets some voltage gated calcium channels. They sense this change in the voltage due to the action potential. The calcium enters the cell. The calcium is responsible for the fusion of the presynaptic vesicle containing the neurotransmitter with the presynaptic membrane. The neurotransmitter travels in the synaptic cleft, and then it binds to specific channels in the postsynaptic membrane. It causes either an excitatory or an inhibitory postsynaptic potential. And at some point, it's going to reach the threshold value to fire another action potential at the postsynaptic neuron, which is going to go to muscles and glands via the ventral root, or it might have an interneuron in between, or many interneurons that are going to integrate this signal, because these interneurons might want to turn. So this interneuron, this one is I'm sorry, why do I have this one? I'm sorry. This an excitatory postsynaptic potential. So it releases acetylcholine, and this has sodium-gated channels. However, this one, interneuron, might want to convert this excitatory into an inhibitory by releasing a different neurotransmitter. Okay, so this is important because the interneurons can modulate the signal. It can increase its strength. Because if one axon gets here in that interneuron and another one of the same one gets here in the same interneuron, then that's going to increase the strength of the output, okay, of that interneuron. So the interneurons are inside the spinal cord, they do not exit, and they're responsible for the modulation of the signal. And again, you have a ventral or anterior root, a dorsal or a posterior root. Now, the organization of nerves, these are the nerves, right? You have the epineurium, the most outside lining. This is connective tissue, of course. Then you have bundles of nerves containing the perineurium, or fascicles, as they're called. Here you can see a fascicle. And this contains both myelinated or, how's it called? Or unmyelinated fibers, but all of them are covered by the endoneurium. Okay. Now, we should talk a little bit more about the reflexes. Reflexes, again, they are involuntary, and they have two major components, a sensory and a motor. The sensory component senses the change. For instance, here you have a receptor in the tendon, and when the tendon is stretched due to the heating of this little tool, as a result, the information travels back. This is a direct reflex, as you can see. You have only one synapse, and it immediately synapses with a motor neuron, and the motor neuron is gonna cause the contraction of the muscle, and hence the elevation of the leg. Here you can see another reflex, a much more complex one. You can see that we have many interneurons, and interneurons that even cross the side. You can see that we have interneurons that convert the information to positive and interneurons that convert the information to negative. These are all excitatory synapses in the beginning from the sensory neuron. However, this one is gonna still do an excitatory synapse because it has the flexor to contract. However, also the extensor needs to be inhibited in order to allow you the contraction. However, in the contralateral leg, the extensor is excited, while the flexor is inhibited in order to make sure that it is held in place. Okay, this is called the crossed extension reflex and the flexor reflex. And you can see that it is a very complex pathway, and this is not even the least. There is such high complexity in those systems. However, you should know that the reflex arc is composed mainly of a sensory neuron and a motor neuron. Sometimes an interneuron, one or more, may be in the middle. And sometimes we can have, always actually, neurons that also send collateral ascending fibers for the purpose of informing the brain about what we just did. Now, the brain has the cerebral hemispheres, two, right and left, and it has lobes, the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe, which are areas of the brain. Now, if we see the brain cut in the middle, we can observe certain structures of the brain. I want to focus on the cortex, which is the most outside. And also this is the brainstem. The brainstem is composed of the midbrain, the pons and the medulla, and also the cerebellum. The cerebellum is important for coordination of muscles and balance. The cerebellum makes the plan for, actually the brain makes the plan for the movement. The cerebellum modifies the plan for your movement. For example, if you want to take a step forward based on the conditions of your body, because you have certain receptors called proprioceptors, which are responsible for informing your brain and your cerebellum for your position at each single moment. If you close your eyes right now, you know if your legs are on the floor, you know how your hand is moving and everything. And that's because of these proprioceptors. They give conscious and also unconscious information about the position, the movement, and everything. So based on that input from the proprioceptors, the cerebellum modifies the movement accordingly. And you can understand that all this information happens very fast, and the transmission through the nerves is very fast, because if we consider a nerve that innervates your foot, for example, that is a very big distance up to your head. Okay, that is more than 1 meter. The axon is going to be almost 1 meter. Again, here you can see the midbrain, the pons and the medulla, and the cerebellum. You can see that in the cerebellum you have the same structure that you have in the brain, with the white matter being in the center and the dark and the gray matter being in the periphery, while in the spinal cord it was vice versa. Another important point I would like to mention is that in the medulla you have the respiratory centers, which are responsible for your respiration. And in the medulla you have also the cardiovascular center. That's why they say injuries to the base of the brain in the posterior segment, so the back of the neck, can be lethal, because you have these important centers there, and if you stop them, it's basically like you stop breathing. You stop your respiratory center from giving a constant order to the diaphragm, say breathe, 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 breathe. And you can have conscious control over it. However, it is mainly a process that happens also unconsciously. Now, just 2 words for the autonomic nervous system. Again, the autonomic nervous system, we said, is controlled mainly by reflexes that happen, like we saw before. However, it can be modulated by higher areas of the brain. The limbic system and the hypothalamus are responsible to initiate autonomic commands. So even though it is mainly by reflexes, the hypothalamus, a very important station for the integration of the autonomic nervous system. Last, you can see the different lobes of our brain, the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. And each of those have different areas responsible for different functions. And here you can see, for example, the frontal lobe is responsible for the mood, motivation, decision making, and different properties are separated in different areas of the brain. And of course, everything is double because we have two hemispheres. Everything happens to the right and to the left. Thank you so much. I know it has been a very long lecture. I have gone in more detail than is required for the IMAT. However, if you are going to get, hopefully you are going to get admitted to medical school, you are you're going to find that information very useful. You're going to find that you're already familiarized with very difficult concepts you're going to encounter in physiology. So not only I'm giving you some information that you're going to need for the IMAT exam, but also I'm going more in depth to ensure that your first years in medical school, your preclinical years, go a little bit easier because you already know some material. Thank you so much for paying attention. If you like our content and you would like to be notified for more uploads, please hit that subscribe button. And if you have any questions, I know it's been a long and difficult and complex lecture, please ask them below. See you in the next video.